I'll be speaking on the topic time to target MET with vision in non-small cell lung cancer. The NCCN and all the recent guidelines recommend to do a broad molecular profiling in advanced non-small cell lung cancer. We know the reason behind. We have number of new mutations that are coming up. And one of these mutations is MET exon 14 skipping mutation. This mutation now has a number of treatment options available. Previously, crizotinib was the only available agent. Now we have capmatinib and tepotinib. In addition, these mutations also give a chance to the patients when we do this testing to be enrolled in appropriate clinical trials whenever available. So the MET exon 14 alterations, they are found in close to 3 to 4% of newly diagnosed advanced non-small cell lung cancers. Predominantly, they are seen in adenocarcinoma histology. There is some suggestion that the sarcomatoid variant has a more frequency of this mutation, and it is the rare patients with squamous cell CA around 2%, and even large cell CA may have this mutation. The oncogenesis with MET exon 14 skipping mutations occurs because this mutation leads to a protein that is lacking the juxtamembrane domain, which is responsible for MET ubiquitization and degradation. So in the absence of this domain, there is increase in the MET receptors on the cell surface as they are not degraded, and thus leading to increased MET signaling, which drives oncogenesis, leading to proliferation, survival, and metastasis by triggering the downstream pathway. In the VISION trial, the MET alteration was tested using the liquid biopsy, tissue biopsy. Particularly, the liquid biopsy was done using the Guardant 360 assay. The tissue biopsy was done using the Oncomine Focus assay. And particularly in Japanese patient, as a trial set, they used the LC Scrum study assay. And in clinical practice, we know that NGS is possibly the best method to detect this mutation. And more importantly, RNA-based NGS is the way to detect this, this mutation. So all NGS, one must remember, is not the same. So there are various studies showing that the NGS platform using DNA will be able to detect only 20% of these mutations, while if we are not doing simultaneous RNA NGS along with it. So the goal is to do both DNA as well as RNA NGS to diagnose this patient. What this study from 2016 showed that this study enrolled patients or looked into the patients from stage 1 to 4 non-small cell lung cancer patients. And it showed that patients who were having MET exon 14 skipping mutation tend to have a worse survival across the whole spectrum of the stage, while the patients without this mutation were doing better. What has also been shown by a number of retrospective studies that patients who are having MET this exon 14 skipping mutation, if they do not receive this MET inhibitor as a part of treatment, their median survival is close to less than a year. While if you use MET inhibitor, their survival is close to 2 years. And it is good to know that now we have a number of agents available for targeting this, from crizotinib, and recently we had capmatinib, and soon in a matter of 1 to 2 weeks, we'll have another agent, tepotinib, available for the use in India on freely basis. At present, the drug was available in as a part on compassionate basis. So the tepotinib at a dose of 500 milligram once daily has a potential to improve compliance. It has a long and a half-life that ensures small peak and trough variations over 24 hours, potentially reducing the toxicity at the peak and suboptimal inhibition at trough. And the VISION trial was the largest study of its kind, which across the globe included close to or screened 8,000 patients with close to 350 patients selected. And these presentations have been done in a number of conferences with a number of international publications. So the vision was a phase 2 multi-cohort, multi-centric trial of tepotinib in patients with stage 3B4 non-small cell lung cancer, patients with PS0/1 with MET exon 14 skipping alteration being diagnosed on liquid or tissue biopsy in a central lab were enrolled. So initially, the trial began in the cohort A in September 2016 when the drug showed activity. Subsequently, a cohort C was enrolled, which was done to assess the confirmatory analysis in this whole population. And cohort B, I won't be discussing, that is for MET amplification. So overall, if we look into the both cohorts A and C, there were close to 275 patients. Approximately 137 patients were treatment naive and close to 148 patients were previously treated. And the primary endpoint was overall response rate, and these were the secondary endpoints. The initial data that I'll discuss now is at a data cutoff date on February 2021. And the trial was designed keeping the clinical practice in mind, because we know that often doing tissue biopsy is not feasible in all patients with lung cancer, where tissue is always an issue. So liquid biopsy was enrolled as a treatment plan. So Vision protocol had 3 analysis sets for all endpoint: tissue biopsy population, liquid biopsy population, and combined population, including both tissue and or liquid biopsy. In this trial, if we compare to other targeted subset, the targeted mutation population, in this trial, the median age was close to 72 years. Patients were elderly. The sex distribution was equally distributed between male and female. The majority, or again, the smoking status was equally distributed. And almost majority of the patients were adenocarcinoma, with around 10% patients having a squamous histology, and close to 20% patients with brain metastasis were enrolled. If we look into the data of patients who responded to the data for treatment naive patients enrolled based on the tissue biopsy, close to 86 patients, the overall response rate was 54.7%. The disease control rate was 80.2%. And it is encouraging to note that almost greater than 90% of these patients had some tumor shrinkage. The duration of this response was close to 32.7 months. The PFS was 15.3 months, and median overall survival was close to 30 months. The patients who were enrolled in this trial, who were previously treated, they also derived numerically a slightly lower benefit, with the response rate of close to 50%, disease control rate of 80%. And also in this previously treated population, also greater than 90% patients had tumor shrinkage. The duration of response was 10.1 months. The median PFS was 11.1 months. The overall survival was 22.3 months. What this data showed that this is the consistent sensitivity to tepotinib therapy regardless of the biopsy method. So whether we do a tissue biopsy or we do a liquid biopsy, whether a patient is treatment naive or previously treated, but what we can see from this table is patients with liquid biopsy were having a lower PFS, whether they were previously treated or they were treatment naive. And possible reason for this was the patients with liquid biopsy had a higher tumor load, and more patients with brain metastasis were there in the liquid biopsy cohort. And recently in ESMO 2022, one of the data regarding efficacy of tepotinib in patient previously treated, and it showed that irrespective of the prior treatment, so whether a patient has received chemotherapy, IO plus chemotherapy, or IO, the duration of response, PFS and OS was seen irrespective of the previous treatment received. If we look into the Asian subset, which was close to 1/3 of this study population, The patients in the Asian cohort were majority were males. Close to 42% were having a smoking history, and nearly 2/3 were previously treated. And again, the benefit, if we look into the PFS and OS data as the numbers what we have seen previously, it is comparable to what was seen in the overall population. Tepotinib was also in this data set, there were 51 patients who are having a brain metastasis, and the overall response rate was 53%. The disease control rate was 76.5%. The median duration of response was close to 9 months. The median PFS was close to 8.5 months. And overall, 87% intracranial disease control rate was seen. And as already discussed, that the vision cohort C was the confirmatory analysis for this data set, and this was the update at 9 months follow-up was presented at WCLC. The cohort C showed that this data shows that the overall response rate, median PFS, median overall survival in cohort C, the confirmatory data set was equivalent to the cohort A, and the median PFS, OS were comparable. If we look into the patients who were treatment naive, enrolled based on tissue biopsy, if we look into the overall response rate in cohort C, again the response rate was close to 62%. The median duration of response in these patients who were treatment naive and enrolled based on tissue biopsy was not reached, while in cohort A it was 46.4 months. If we look overall, including cohort A and C, the median duration of response was close to 46 months, while the median PFS was approximately 15 months, and median overall survival was close to 2 years. And also similar benefit in cohort C was seen in patients who were previously treated and enrolled based on tissue biopsy. So median PFS was close to a year, and median overall survival ranged from 19 to 20 months. In the cohort C also, the duration, the overall response rate was equivalent to what was seen in cohort A. The overall efficacy in cohort C and A was robust and durable across therapy lines. So whether it was first line based on tissue biopsy or liquid biopsy, or in second line, the median PFS in the cohort C was slightly better, and the median overall survival was similar. The tepotinib in general, if we look into the tolerability, the discontinuation rate, if we look into the both cohorts A and C, it was close, the permanent discontinuation was close to 15%. The most common side effect was any grade was peripheral edema, and this was also the most common grade 3 side effect. The grade 3 side effect subsequently more common was hypoalbuminemia and ALT increase, while other side effect like blood creatinine increase, which is possibly a class side effect, which is also seen with capmatinib, was greater than grade 3, was less than 1%. So the simple once daily regime of tepotinib improves the patient compliance, as already mentioned, less than 15% discontinuation, adverse profile which can be managed by simple dose modification, low treatment related peripheral edema, and the other side effects as mentioned previously. And this safety profile has been tested in approximately 300 patients in this vision trial. And in metastatic setting, one of the goals of our treatment is quality of life. And as mentioned in the vision trial, the median age was close to 72 months. So even in these patients, if we look, the quality of life was maintained. If we look into the comparison between tepotinib and capmatinib, we know we cannot compare the cross-trial data because patient population may be different. But what is encouraging to note that both the drugs are active with a good more than 50% patients in the first line treatment being having a response, and the median of duration of response with tepotinib in the treatment naive cohort was 32.7 months. Again, disease control rate, median PFS is almost comparable, and grade 3 or greater side effect was slightly means lower in the tepotinib as compared to capmatinib. So tepotinib with simple once daily dosing is one of the drugs which we can use in these 3 to 4% patients which are having otherwise poorer prognosis, which will improve, which will lead to the efficacy endpoints as discussed. And also we have now data based on the vision that we can very confidently give it either based on tissue or liquid biopsy, knowing that the safety is good and also the quality of life is maintained. Thank you.
There is a question for Dr. Shukla, I think, regarding the liquid biopsy. Someone has asked. So, will you go in the question and answer box if you would like to answer it?
Sir, will you use this drug for MET amplification? So
that is my question, but there is another question in question and answer box also. Not in the chat box.
Okay. Is it liquid biopsy is gold standard? Liquid biopsy is diagnostic test or monitoring test for those patients who has blocks not having any tumor content. So liquid biopsy for MET exon 14 skipping mutation can be used as a diagnostic test. Recently, we have data for EGFR based on the APPL data. We can monitor and change treatment in EGFR based on the liquid biopsy. But for MET, I don't think we have data that we can change treatment based on the liquid biopsy. It is more of a diagnostic tool at present for patients who are having no tissue, means we can use this simultaneously along with tissue biopsy.
And would you like to use it, Dr. Shukla, as on today for the amplification also, or it's only for MET exon 14?
So, sir, again, means the approval as such, if we look, is for MET exon 14 skipping mutation. But if we look into the data for the cohort B, the data is almost as good, I think, means the PFS is similar, though slightly lower, but I'll be using it even in MET amplification.
Thank you.
Thank you, sir.